DNA digestion, troubleshooting and complex scenarios. Troubleshooting a failed digest is a rite of passage. Success requires a systematic look at your substrate, enzymes, and reaction environment. Some solutions are quick fixes, others demand more time or specialized reagents. Let's start with the quick fixes: purity, conditions, and cofactors. If your 260-230 ratio is below 2 or 2.2, contaminants are inhibiting your reaction. Don't waste time. Perform a cleanup before moving forward. Next, never assume 37 degrees Celsius is the standard. Always verify the optimal temperature of your enzyme on the technical data sheet. Additionally, watch out for the gel shift effect. This occurs when an enzyme remains bound to the DNA, making fragments appear larger or smeared. As you can see in this example from Thermo Fisher, using a loading buffer containing SDS before running your sample denatures the enzyme and releases it from the DNA, ensuring your bands reflect their true size. Also, remember your cofactors. If your enzyme requires cofactors like DTT, ATP, or SAM, use fresh aliquots, since these reagents degrade quickly and are often the hidden cause of failure. Finally, flick and pulse spin your enzyme tube to resuspend it. Skim the surface when pipetting, to avoid transferring excess glycerol into your reaction. Now let's tackle complex topologies and sequences. Supercoiled DNA, hairpins, and cruciforms can all hinder cutting efficiency. If you're seeing incomplete digestion, start with the simplest fix: increase your enzyme units and extend the incubation time. You can also try linearizing the plasmid to release torsional strain, or heating your DNA to 75 degrees Celsius to open the structures. Just remember to snap cool it on ice before adding the enzyme. Finally, remember that GC content dictates efficiency. Some enzymes favor high GC, while others prefer low. If the balance is off, compensate by boosting the enzyme concentration and giving the reaction more time. Don't forget DNA methylation. Some enzymes are blocked by methyl groups added by standard E. coli strains. If you're hit by this, you have two options: switch to an isoschizomer that is not methylation sensitive, or retransform your plasmid into a DAM DCM deficient strain to obtain unmethylated DNA. Finally, watch out for multi-site enzymes. Certain enzymes require two recognition sites to function, one for binding and one for cleavage. Think of that second site as a necessary cofactor. If your template lacks the extra site, supplement the reaction with double-stranded oligonucleotides containing the recognition sequence. Start with a 4:1 oligo to plasmid ratio to get the reaction moving. And now let's look at some shortcuts. If you're managing incompatible buffers or temperatures but don't have time for full sequential digestion, you have two workarounds. First, start with the low-salt reaction. Once it's finished, simply add the missing salts or preferred buffer and your second enzyme directly to the tube. Alternatively, if an enzyme has at least 50% activity in the other's buffer, increase the units and extend the incubation time. In either case, keep the total glycerol under 10% to prevent star activity from ruining your results. And for some tips for traditional cloning. If your vector and insert don't share identical sites, look for compatible sticky ends. Using different enzymes that leave identical overhangs can solve the problem, but always verify your in silico map first. This creates a scar that can cause frame shifts or unwanted mutations in the coding region. Lastly, if your sites are too close together, steric hindrance may prevent cutting. Enzymes typically need 6 to 10 base pairs of flanking DNA to dock. To fix this, clone a double-stranded oligonucleotide stuffer to provide the necessary spacing. If all fails, sequence the template. Sometimes the problem is a mutation that has physically destroyed your recognition sequence. Troubleshooting traditional digestion is a grind, but it's an essential skill for any molecular biologist. In our next video, we're moving beyond the basics to modular cloning. I'll introduce you to how to employ Golden Gate assembly to build complex multigene plasmids from single functional units.